to kind of see new faces and see old friends too. Kareem and I have interacted with each other at various meetings along the way. Typically, I don't know, I guess more kind of the geochemistry, kind of geomicrobiology type meetings. So what I want to do today is tell you guys a few different stories. Some stuff that we published, some stuff that we're working on right now as well. And I'll focus mostly on the iron reduction side of things, but tell you some new stories where we've moved away from our main model system into some others. And we're learning that not all microbes reduce metals for the same purpose, I think. And I'll explain that as we go. And then I'll end with some of the work that actually was just published, I think, yesterday, where we've been doing some genetic manipulation of iron oxidizing bacteria. So just a quick kind of general overview. I want to start with this little microbe that we know and love called Shewanella. I'll give you a short introduction to that microbe and talk about this process called extracellular electron transfer, and how we've been exploring this process in some of Shewanella's relatives, other gamma proteobacteria. And then how we've leveraged our findings kind of based in fundamental genetic work of actually demonstrating that genes that we find in these microbes are correlated to these iron reduction activities that we quantify in the lab. And we take the genetic information and then began moving out into sequence space. And as I'm sure many of you are familiar with, maybe you're not super familiar, but it basically costs almost nothing to sequence a microbial genome these days. I mean, literally $50 can get you a microbial genome. And as a consequence, the databases of genetic information in microbes has just exploded over the last 10 years. And so what we've done is we've taken some of these genetic constraints, these kind of lab-based experiments, and then moved into that sequence space to look at the distribution of extracellular electron transfer. And we've got what we think is really good evidence for at least in some cases this metabolic process that facilitates iron reduction in the environment and maybe iron oxidation, how it can move around in the bacterial world, and I think that's really interesting. And then finally, we'll talk a little bit about using genetic methods, or the more fancy way of saying it is synthetic biology, to understand extracellular electron uptake in iron oxidizing bacteria. And so I think, as I mentioned a little bit ago, and for people that maybe have just popped in, if you have questions along the way, feel free to interrupt. I do have the little chat window up if you want to throw in a chat. Otherwise, I'll be happy to answer questions at the end too. Okay. So let me introduce you to this little microbe called Shewanella. I like to call it the E. coli of the environment. And I do this for a couple of reasons. One is E. coli is really easy to grow, and as is Shewanella. We know a ton about E. coli because of how easy it is to grow and how quickly you can do experiments in it. It's one of the reasons I really like Shewanella, although some other very interesting microbes that are more difficult to grow, as you'll see at the end, we've been trying to kind of come to grips with that. But so these bacteria are widely distributed in the environment, mostly in aquatic systems, mostly marine, although they can be found in freshwater systems. And their wide distribution, we think, is driven by two kind of basic properties of these bacteria. One is they have a wide temperature range at which they can grow, and so most of them are capable of growing at kind of at mesophilic temperatures, but a lot of them can also grow at very low temperatures. A handful are true psychrophiles, but most of them just have a wide temperature range. And as you can see, many of these arrows are pointing to cold environments, and I would include these ocean ones, and certainly the North Atlantic and the Antarctic as these. The one that we know the most about is called Shewanella oneidensis, and that's the one that I'll kind of frame much of the work today. That one comes from a lake in upstate New York called Lake Oneida. So it's a freshwater isolate, although it does require salt for optimum growth, and so we think that it's probably been in that lake for probably some sort of short period of time. I don't know if that means a few dozen years or a few hundred years or 1,000 years. I don't know exactly what that means, but we think that ultimately it had a marine origin. And then we've also studied one in the lab called Shewanella gopherei. This one comes from, so this is pointing more or less to Minnesota here. And this comes from the Mississippi River behind our student union on the Minneapolis campus. that has a lot of similar properties. So you can find these guys just about everywhere. They're very easy to grow, which is one reason why you can find them easily. So the other reason that they are widespread, and here's a couple pictures of my kids that may interrupt and may not. I don't know. They just ran by the door. So they have this wide temperature range at which they can grow, but they also can breathe lots and lots of different things. And what I mean by that is they have an ability to take electrons from the food that they eat, and what we do is we're taking those electrons and putting them onto oxygen. These bacteria can do that as well, but then if oxygen's not around, they can put them onto lots and lots of different substrates. And the Shewanellas are known to be the most diverse respiratory organisms that have been described so far. Over 30 different compounds are known to be respired by these bacteria. Many of those compounds go through this one pathway that I'll talk about, that is this metal reduction pathway. Okay, so I have twins, so I have to show you a picture of my son too. That's Logan. Also converting donut into CO2, protons, and electrons. And even graduate students have this metabolism. This is Bridget. I'm going to talk a bunch about her work in Aeromonas and Vibrio a little bit later. Okay, so here's just a partial list of the respiratory diversity of what Shewanellas are known to be able to respire. So oxygen is on this list, and maybe a few of these things you're familiar with, and probably a few of these things you guys are familiar with. But what really sets these guys apart, and what made kind of their discovery back in the late 1980s, along kind of the parallel discovery of Geobacter with this metabolism, is their ability to respire iron and manganese, which at circumneutral pH in our aerobic world are insoluble oxide minerals. And so these are just some images of a fluorescently labeled Shewanella. So you can see these little fluorescent cells here, using iron oxide as a terminal electron acceptor, and manganese oxide as a terminal electron acceptor. So this was the first kind of respiration described where the substrate was not coming into the cell, but rather the electrons were being brought to the substrate outside of that cell. And it had gone through various different terminology in terms of how to describe it, but I think in the last maybe 10 years or so, maybe 15 years, we really settled on this process called extracellular electron transfer, which in this case, we're talking about electrons from inside of the cell going out. So why does this matter? Well, the redox state of metals almost always influences their solubility, toxicity, or both. In the case of iron and manganese there, when bacteria put electrons onto it and reduce those substrates, they become soluble in their reduced state. In the case of something like arsenate going to arsenite, you take something bad into something worse when you reduce it. In the case of uranium, you can take something that in its oxidized form can move pretty freely in groundwater systems, but when it becomes reduced, its mobility dramatically decreases. And so trying to understand this from a fundamental level is important in understanding the mobility of redox active metals in the environment. And from my perspective, so I think all that stuff is cool, but really from my perspective, thinking about microbes and trying to understand how they do things, the respiration of these insoluble substrates requires very unusual or atypical electron transfer pathways compared to, say, how we breathe oxygen in our using proteins in our mitochondria, or how a microbe might breathe something like nitrate or DMSO. Because these bacteria can move electrons to the outside of the cell, we can replace the iron in this system or the manganese in this system with poised electrodes. And getting rid of the metals ends up, so using these electrochemical devices represents their own challenges, but getting rid of the metals really allows us to look at the physiology of these bacteria in terms of electron flow in a very specific way. And there are some commercialization opportunities kind of in this area. People will use things called microbial fuel cells to generate small amounts of current, say in a wastewater treatment plant, or to power devices at the bottom of the ocean. And so again, our work, I guess, is related to that, although we don't do any of that direct work. We're trying to understand the fundamentals of how the process works. It could also be bidirectional, meaning that electrons, redox pathways in general can move both directions, provided the potential is correct. But I'm not really convinced that Shewanella brings electrons in through this pathway that I'm going to describe. naturally, although there's been some recent work in this area, so I'm still trying to digest that. Okay, so how does this work? I'm not going to go through the genetics, and a lot of this is stuff that everybody in the field agrees on. We disagree on some other details about this, but that I'm not going to go into. But in general, so these are gram-negative bacteria like E. coli. So they've got two membranes to deal with. They have the cytoplasmic membrane or inner membrane, and it's across this membrane that the bacteria would be making a proton gradient that would drive things like ATP synthesis and motility, for that matter, and transport of certain nutrients. And then there's an outer membrane here. And in order to get electrons out from the quinone pool in this membrane all the way to the outside of the cell requires all these proteins in red and this protein in gray here too. So the red proteins are all multi-heme cytochromes, and the heme groups within those proteins are basically the electron carrying cofactors in the system. All these three down here each have 4 heme groups. MtrA here has got 10. MtrC also has 10. And MtrC looks like if electrons can get in one way and come out a couple of different ways, which again gets into some of the more subtleties of extracellular electron transfer in Shewanella and things that we start debating in the field. This is a crystal structure solved almost 10 years ago by Tom Clark. Of a paralog of this protein, and you can see the iron atoms are these orange balls at the center, and you can see there's 10 of these iron atoms coordinated by these heme molecules, and that electrons can come in basically at the bottom of this protein, and they can probably go out here in this red domain, out here near this green domain, and out through the top of the protein as well. And again, you can ask me questions about this later, but we think that through the wings of this protein would be mediated by electron shuttling and redox active shuttles that these bacteria produce. And then through the top of this protein, we think is direct electron transfer. We have, we're just starting a series of experiments to try to begin unraveling that. And hopefully we'll be able to collaborate with Tom and his group to actually solve some structures here along the way, but we're a little ways away from that. So just earlier this year, sorry, last year, whatever year it is. So shortly after the pandemic began, Marcus Edwards, who was a postdoc with Tom Clark, solved the structure of this entire complex here. And this is really impressive because it allows us to see this protein and this protein complex that is a conduit to allow electrons to go from the periplasm of the cell across this outer membrane. And this is a motif that we think is conserved pretty widely. And I'll show you some evidence for that in some of the genomic stuff that we're going to talk about. But even microbes like Geobacter have got proteins that look kind of like this protein here, this MtrB. So it would be a kind of a beta barrel domain, proteins that have multi-heme cytochromes on either side of predicted to be on either side. So we think that this is probably a conserved strategy that gram-negative bacteria have anyway, to be able to move electrons across their outer membrane. And so it's one of the we're basically, as we surf around and look for this capacity in other organisms, looking at the databases, this is one of the things that we're going to be looking for directly. Okay. So there are lots and lots of microbes that have been reported to be capable of metal reduction. So this is just a partial tree of kind of the microbial world. And just to orient you a little bit, Shewanella falls here in the gamma proteobacteria. The other one that people study is called Geobacter. And this one is down here in the delta proteobacteria. These guys, they do things differently, although they do use multi-heme cytochromes to accomplish this electron transfer business. But Geobacter is much more complicated than Shewanella. Again, you can ask me questions about that later if you want. But that's probably because Geobacter really does metal reduction for a living, and Shewanella probably does it to get by. It would prefer to do other things. But Geobacter, I think, really does kind of has evolved to really do this for a living. Okay. But there's other lightning bolts all over the place. So that people have either demonstrated or suggested that that this capacity to move electrons to the outside of the cell has been observed in lots of different organisms. So I'm going to zoom in a little bit to the gamma proteobacteria. So these are the relatives of Shewanella. So Shewanellas fall here. And I'm going to tell you a story about a Vibrio and a story about an Aeromonas. The Aeromonas falls here inside the Pseudomonas clade. I think we're going to do Aeromonas first. Or maybe I'm going to show you, let me show you what these gene clusters look like first. So in the Shewanella genome, all of those components that I showed, these guys in the red are form that outer membrane conduit. And then elsewhere in the genome, you've got these other components that take electrons from the cytoplasmic membrane and move them into the periplasm. So those would be the blue and the green components. In Aeromonas hydrophila, they have a very similar outer membrane conduit here, as does Vibrio natriagens. And I'll introduce a little bit more about Vibrio in a second here. In each of these other cases, they use slightly different strategies to deliver electrons to that conduit, but the conduit is highly conserved. So let's just look at between the conduit protein, or the genes that encode the conduit proteins from Shewanella versus this Aeromonas strain. And this is a percent identity. So the last protein here, this would be the beta barrel protein, is 67% identical. These guys have pretty high identities as well, although this is kind of at the borderline. Looking across the different Aeromonas strains that have been sequenced, the little lightning bolts represent where we see this pathway. And most of the hydrophila isolates, of which at the time that we did this, which was a few years ago, there were 41 complete genomes of hydrophila isolates. Almost all of them had this pathway. It doesn't, other than being dominant in hydrophila, it doesn't seem to be consistently found across the species, right? You see just a few little lightning bolts here and there. Here's a Veronii, which is also highly sequenced, and just one of those isolates seem to have this. So this is not like, it doesn't seem to be a common property of Aeromonas strains. And this was kind of our first hint that maybe this capacity might be moving around out in the environment. So let me show you just a little bit of data to demonstrate that that at least one of these genes is essential for the process of iron reduction. So it's one thing to gaze at genomes and make predictions about what things might be doing, but it's always nice to be able to demonstrate that that pathway really is required for the process that you think it's involved in. And so what I'm showing you here are metal reduction curves looking at iron citrate or chelated form of iron 3, iron oxide in the middle, and manganese oxide on the right, where we quantify over time either iron 2 production or manganese 2 production. And in pink, you have the wild type strain reducing over time. And you can see here, this is iron citrate goes fast. It's soluble, and so cells do this reaction pretty fast. But for iron and for manganese, this is now a couple of weeks. So this is about twice as slow as Shewanella, and that's something we'll come back to a little bit later. But what you see in pink there is the wild type reducing producing iron 2 over time, and manganese 2 over time. In blue is where we've gone in and we've deleted one of these genes. In this case, we deleted the MtrA. MtrA is from the big conduit thing, whoops, is this protein here, or the one that's really kind of buried inside of B. And without this component, the system can't work. We know that from Shewanella work. Okay, so we've deleted that, and you see this activity, the iron reduction activity goes away, the manganese reduction activity goes away. And then we can complement that by meaning that we basically can fix that defect by providing a good copy of that gene back to the cells. The other thing that we can do is we can take parts of Aeromonas and move them into Shewanella. And the reason that we do some of that work is to just verify that these components really are doing what we think they're doing. So now we're looking at iron citrate reduction. Now, if you look at the time frame, it is quite faster. So now this is basically done in just a few hours when we do this experiment with Shewanella in pink. In blue is a strain of Shewanella where we've removed a whole bunch of things from the genome. So it really has no capacity to do metal reduction anymore. And then when we put back this cluster, MtrC, MtrA, and MtrB from Aeromonas, we restore that iron reduction activity. So we know that these Aeromonas components can replace the Shewanella components here too. And when we do that, it goes just as fast as wild type, right? Or almost as fast, pretty fast. Okay, so let's move over to Vibrio, and we've done a whole bunch more work, kind of defining all of these other bits and pieces, but I'm not going to get into it today. We published this paper a couple of years ago. If you're curious, or you can ask me about it. All right, so now we'll switch over to Vibrio natriagens. So we see this pathway in a bunch of different Vibrio species: Vibrio parahemolyticus, Vibrio vulnificus, Vibrio diabolicus. There are a boatload of Vibrio cholera strains that have been sequenced out in the database, and we've never seen this pathway in any of the choleras, which is, I think, kind of interesting. So here we have the Shewanella pathway at the bottom, and now we have percent identity. This is back at the Aeromonas one, and now here's Vibrio natriagens up at the top. Or these are percentages of Vibrio natriagens to each of these other ones. So you can see this pathway here. MtrC, A, and B from Vibrio natriagens is pretty similar to the pathway here in Aeromonas, and also fairly similar to Shewanella. Vibrio natriagens is an unusual microbe in that it's the fastest growing organism that people have ever identified. So it has a doubling speed of around 9 minutes. If you give it like an obscene amount of nutrients in liquid medium and grow it aerobically, it grows incredibly fast. It does not do metal reduction incredibly fast, as we'll see in a few seconds here. But we ended up using this as many years ago. We had a parahemolyticus strain in the lab that we tried to do some of these experiments with, and we could never convince ourselves that that strain was doing metal reduction. But with this natriagen strains, we did figure out some conditions where we could see metal reduction occurring. And so here's a little bit of data from our recent paper that came out just a few months ago. So here we have, we're looking at iron citrate reduction. I should have written that. Oh yeah, I did write it up here. Iron citrate reduction over time. So you can see right off the bat that this is pretty slow. So here's the wild type Vibrio strain in red. And now instead of hours, this is days. If we go in and delete the genes encoding MtrCAB, these red guys up here, we now substantially decrease the amount of iron that is reduced in that same time frame versus no cells at the bottom here. So that's convincing data that these genes are involved in the iron reduction process that I'm describing here. Iron reduction in Vibrio is a little bit strange in that we don't actually know what they're eating, and I'll explain that. I won't go into huge detail, but I'll just say we basically we give them galactose, which they ferment, and then after they're done growing and done fermenting the galactose, then they begin doing iron reduction. And we're not exactly sure what the byproduct is. And we tried a bunch of different things on its own, you know, things that we were guessing could be electron donors, and we couldn't recover the same amount of iron reduction activity. So that they're making some sort of substrate or substrates as a byproduct of fermentation that then they're using to do metal reduction. So it's a little different from what Shewanella or Aeromonas can do, where we actually know what the electron donor is in those reactions. So here, now we can do the same experiment taking this JG1453 strain. This is the strain of Shewanella where we've deleted all these various metal reduction components. And we can put MtrCAB back just from Vibrio. And so here's wild type Shewanella. Now, instead of days, we're looking at hours because this goes quite fast in Shewanella. And when we put that conduit from Vibrio into Shewanella, it goes just as fast too. So even though in this experiment, we're looking at days for iron citrate to get reduced, when we take this pathway, at least that component from Vibrio and put it into Shewanella, we know that these components can go as fast as Shewanella does. So this has set up some interesting observations that we don't have any answers to this yet, but we have some ideas, and hopefully we'll be able to explore these in the next kind of couple of years. But what I'm showing you here are the various pathways for how electrons get to the outside of the cell in Shewanella, in Aeromonas, and in Vibrio. And you can see here that Vibrio is almost an amalgamation of some Shewanella. So they all share the same outer membrane components, right? That's all the red components there. Um, the blue components in the inner membrane, in Aeromonas, it's a totally funky system. Again, we could talk about this if you want to, but they've essentially evolved nitrite components from nitrite respiration to serve specifically for metal reduction in Aeromonas. So they have elsewhere on the chromosome like an authentic nitrite respiration pathway, but these ones are located just downstream of these other genes. But in Vibrio and in Shewanella, they use the Cyme-like protein. Both Aeromonas and Vibrio use this little this little protein here called PDSA, which is a diheme cytochrome, rather than in Shewanella that is using tetraheme cytochromes. And so we don't know why the speed is different. We know that it's not something fundamental about what this complex can do, because we can take the complexes from Aeromonas and Vibrio and swap them into Shewanella, and they go just as fast as Shewanella does. So that tells us it's something about this pathway of how electrons are sent to it. Or it's something about the core metabolism, which is the ultimate source of these electrons. Remember, all these electrons are from the food that these bacteria are eating. Or Shewanella is just really good at making multi-heme cytochromes, and these other guys, Aeromonas is okay at it, and Vibrio just kind of sucks at it. And we don't really know the answer there, but like all those possibilities are on the table. And the interesting consequence is that, and I'll show you one little bit of data from Vibrio here. Shewanella and Aeromonas can grow with this metabolism, so they can double slowly, but they can with metals as an electron acceptor for anaerobic growth. Vibrio doesn't seem to be able to grow with this metabolism, but rather, so why do it? We think it might benefit these guys when basically they have nothing else to do. So we think it enhances their survival, and the little bit of data that we have to suggest this is by simply growing these cells up. So what we're looking at here are colony forming units over time. So we're quantifying viable cells in these cultures over time. And what you see here is these guys are growing up, and then they're persisting a little bit, and then they're dying over time. And the rate at which our mutants die is enhanced compared to wild type. So wild type here is able to reduce the citrate that we provide. And we can see this here, quantifying iron 2 over time. We see a bunch of iron 2 coming away. And these guys persist better for a longer period of time. Eventually they crash too. But we think that under these kind of more starvation conditions, their ability to move electrons, even though it's slow, to this insoluble electron acceptor allows them to persist longer. And these two mutants, one is a mutant taking away those things, and one is a mutant taking away this, and both of them basically behave the same. And this didn't seem to be just a simple nutritional effect. So if we just give the cells a bunch of extra iron 2, we don't see a very significant difference here between survival. In fact, that much iron 2 seemed to be more detrimental to them. They actually are lower colony forming units. There are fewer viable cells in that experiment. And it could be that as we explore looking for this pathway in a bunch of other organisms, I think in maybe most of the cases, this is not going to be a respiratory process, but rather it might be something related to survival. The other thing that we're thinking about too is that it might actually be related to nutrient acquisition. And by putting electrons onto iron oxides and causing that iron oxide to start dissolving, you can mobilize things like phosphate, which could be quite limiting in the ecosystem. Or maybe what you really want is that iron. And this could be a strategy to mobilize some iron that you would need for core metabolism, and not just as a respiratory substrate. And maybe that explains why they just don't do it so fast, because they don't need to. Again, we don't have any answers there, but those are things that we're thinking of. Okay, so we're going to take learning from Shewanella and Vibrio and from Aeromonas, we can now begin exploring where else we see this capacity in the sequence database. And for that, this is work that's been driven by my grad student Bridget, and also Izzy Baker, who's a grad student with Pete Gerges at Harvard, who's a collaborator on this project. And so what Bridget and Izzy did is they took the MtrA ABC sequence from Shewanella and made and concatenated it. So they stitched it together, and then searched the database for all of these for this entire set. So we constrained the search based on finding those genes next to each other in the same order, and then began looking at the organisms that we found. So people have done this search before, say looking for one or the other, but we made this decision that we wanted to look for this entire conduit. So there is Marcus and Tom's image again of MtrA, MtrB, and MtrC on the top. And what we saw are 148 species, 13 orders, 5 classes, all and 3 different phyla, all gram-negative, as you might expect, because the gram-negatives have the outer membrane where this conduit would be. And so, not surprisingly, so like kind of all of this stuff here, and again, you're not supposed to be able to, I can't even read that. You're not going to read that. But these are all the Shewanellas and a couple other kind of Shewanella-like pathways. The Aeromonas and the Vibrios are all kind of here. And where it gets funky is all of this stuff here. And I just want to zoom in on clade 3 and just show you something here. Oh, actually, before I do that, for some of these genomes, we have information about where the strain was located from. And so this is without any of the Shewanella data. Otherwise, the Shewanella dots, there's so many of them, they just overtake the map. But it kind of gives you a flavor for where we find organisms with this predicted capacity to move electrons outside of the cell. Marine systems, so like from animal or associated with algae. So that would be maybe like a fish or a squid isolate or something. In marine sediments, all sorts of different freshwater systems, glacial sediment, non-marine saline and alkaline places, so brackish and salt lakes and soda lakes. And then we have this kind of, I like this being engineered / medical, so oil, wastewater, bioreactors. So those would be like enriched in, say, a microbial fuel cell, or even some clinical isolates too. But you can see they're all over the place. So there's not necessarily a rhyme or reason for why a microbe, based on its location, might have this capacity. So, but I think it's interesting to start looking at. Okay, so let's zoom in on this third clade. And the reason I wanted to show this one in particular is that there's some funny things going on here. Basically, what's happened is we see evidence for what we think is horizontal gene transfer, because we end up having organisms from a whole bunch of different groups of microbes. So we've got alpha proteobacteria, beta proteobacteria, gammas, and there's a gemma in here too, that all have pathways, MtrCAB pathways, that are phylogenetically pretty similar. And yet, if we run a tree looking at, say, the 16S gene of these microbes, or look at the true phylogeny of these bacteria, they're not related. And so this is an instance where what people would call phylogenetic incongruency, where the tree drawn, the relatedness of these bacteria drawn by using these genes is different from using kind of the genes that we would use to categorize these bacteria. And so that began Bridget and Izzy kind of diving in and looking at some genomes, and you end up with stuff like this. And this is one of those slides where they say, I know this is complicated, but let me walk you through it. I'm not going to do that. It's just a joke. But this is where we begin comparing genomes. And then we zoom in to look at specific regions where the genes that we care about are. So here's a couple of examples I just want to show you. So here we've got the MtrCAB pathway in blue. And there's little PDSA and Cymase. So this is a complete pathway that will allow electrons to go, again, based on our work in Vibrio and Aeromonas, would allow electrons to go from the cytoplasmic membrane all the way to the outside of the cell. And we have a very closely related Marinobacter strain here, which is actually the strain that our colleague Sarah Glavin works on, called Marinobacter atlanticus CP1. And you can see the exact spot where these genes are different in this Marinobacter isolate versus this one. It is a little interesting in that heme biosynthesis is located, or at least part of it is located right here, which is essential for production of this one, of 4 out of 5 of these genes. So there's one instance where we have 2 very closely related bacteria. One has this capacity and one doesn't, and we can see right in the genome where it's different. These gray bars here are showing you how similar things are. So the darker this is, the more similar it is. So the light region here would be just kind of similar. The darker regions are more similar. So that's showing you here percent identity. Another instance, so these are now 2 different Marinobacter strains, also being compared to Sarah Glavin's strain, CP1. This W62 strain has got this pathway here again. And then there's another Marinobacter strain that has got this exact same pathway with a whole bunch of extra bits here. And then this region in the CP1 strain is littered with these elements that are often correlated to horizontal gene transfer events or transposition events. So you've got a bunch of these annotated transposase and integrases and nucleases. So this is a region of the CP1 genome that is probably changing. And our guess is that this strain probably lost this capacity over time. So a couple other instances here, again, where we see two closely related organisms that either have or don't have this pathway. Thioalkyl Vibrio is one here. This one I really like because in this strain, this ARHD1 strain versus Sulfitophilus, you have these MTR components. Now, these are just the outer membrane components. It's not clear what the periplasmic and intermembrane components are, but you also have an intact cytochrome maturation pathway right here that you need in order to make A and C. And so if this entire piece moves around, you might have that entire metabolic capability, not only of the genes that you need to make this pathway work, but also the process that you need to make the cofactors to make those proteins work. I think that's kind of neat. And then we even have, if we dig around in some Shewanellas, so most Shewanellas have the capacity to respire metals, but not all of them. And here is a case where we think is a loss. So here's two closely related Shewanella strains, Halifaxensis and Paleana, that have this MTR cluster here. And the Shewanella MTR clusters, in most cases, are more complex than the Vibrio and the Aeromonas ones. But you can see here, Shewanella violacea, which is a deep sea Shewanella isolate. Those genes just seem to be gone. So we have very similar sequences up and downstream, but those are just gone here. So we think that's a case of it being lost. Okay, so that was some of the work there for the extracellular electron transfer and how it moves around. The last few minutes of the talk, I want to talk about microbes running the opposite reaction and some of our more recent work in iron oxidizers. So we focus mostly on, or we focus so far only on bacteria doing this reaction, taking electrons from inside of the cell and reducing iron 3 to iron 2. But now I want to talk about bacteria running the opposite reaction. And the specific microbe that we're going to talk about is called Mariprofundus ferrooxydans. This is the founding member of the Zeta proteobacteria isolated by Dave Emerson probably 15 or 20 years ago now. I forget exactly. It's an obligate iron oxidizer. So this is the only way that we know of how it can grow either in the lab or in the environment. It's neutrophilic. It's a chemolithoautotrope, so it's using chemical energy. It's using iron as its electron source, and it's fixing CO2 like a plant, essentially, using rubisco. So this is the metabolism. It's taking iron 2 with a little bit of oxygen and making iron oxide. So, and instead of an electron going out of the cell, this electron is coming into the cell. So these guys were isolated from a place called the Loihi Seamount, which is off the coast of the Big Island of Hawaii. And they're working at this chemical gradient where there's iron 2 coming in from a hydrothermal source, mixing with the aerobic seawater above. And these guys are catalyzing iron oxidation at a slightly lower oxygen concentration than is favored abiotically. And we're very interested to know what that pathway looks like. We've got ideas, but so far we don't have any great evidence to support these ideas. Hopefully we'll be working on that in the years to come here. Here's an SEM image of this taken by Clara Chan. So you can see this iron oxide stock made by these little bacteria. Oh, I should point out, almost all the iron that you see here is made by these little bacteria. And then here's a TEM image also taken by Clara. And you can see these guys have two membranes like Shewanella and E. coli, but then they've got these iron oxide filaments coming out of the side of the cell. So what's really fascinating about these bacteria is that they must have a complex. Now imagine the complex that I showed you from Shewanella that we actually know the structure of. Something maybe like that sitting on the outside of the cell, but instead of directing an electron out, it would be taking an electron in and then catalyzing the formation of that iron oxide mineral right at the curved part of this cell. Not all the Mariprofundus isolates do this, but this particular strain, PV1, does this, and it's fascinating, I think. You can see here there's a little flagellum right here. So these guys are still are modal as well. And so that's another thing that we can chat with. A few years ago, the idea was if they had proteins on the outside of the cell that were allowing electrons to come back in, then we should be able to remove iron from the system and instead use a cathode in order to get these guys to grow. And this is just a little bit of data from the paper that we published a few years back, just showing that if we have biology on these electrodes versus no biology, we see some differences in electrochemistry, which suggests that biology is causing the electrons to flow a bit differently. Here we're looking at current over time, and the biology here is taking electrons in, and so we have kind of this steady rate of abiotic oxygen reduction. But when we put the biology in, we see that getting enhanced. Again, it's super slow here, and we can see cells on the surface of these electrodes and whatnot. Again, that's older stuff. So we're excited about this organism, trying to understand it. And again, if we understood what those genes were, there's all sorts of things that we can do with that. And so we began trying to develop some genetic methods. So here's our first success at getting these guys transformed with a plasmid that's shown here. Getting DNA into a cell is kind of the first step at being able to do genetics. This is a big challenge because the bacteria don't form colonies on plates. And so doing genetics is a bit trickier because of that. Once we got DNA in, we could then put genes onto those plasmids. And of course, our first one that we did is a little protein called GFP, or green fluorescent protein. So you can see here now that the little Mariprofundus cell is expressing this fluorescent protein and making this twisted iron stalk. There's a little close-up of it there. This is just a combined light and fluorescence image. We sent the strain over to Clara's group, who can do kind of fancier images. And I like this image because you can see these 4 cells here are the great grand cells of the grandmother cell that began here. So you had a cell here, it oxidized this much iron and then divided, sending the stalks in each direction, oxidized that much iron and then divided again. So this is kind of a neat way to be able to track these bacteria. And Clara made this little movie. This is kind of time lapse over 140 minutes, and you can see the cells kind of twisting as they're oxidizing iron over time. Okay, so we've been so making them glow green is fun, but it doesn't really teach us anything, and doesn't really it can help us do some things using microscopy. But really, what we wanted to do is now we want to try to get these guys to grow better under lab conditions. And we began trying to teach them how to eat fixed carbon. And the baby step in this is to replace the CO2 that they're using to make biomass with fixed carbon. And so we gave them a pathway to bring in xylose, an isomerase, and a kinase to then feed this sugar right into the pentose phosphate pathway. And when we do this, if we take away carbon dioxide, the cells don't grow anymore. We get a little bit of abiotic iron oxidation. But with xylose, we now get these twisted stalks again. So this tells us that this pathway is working. And the same trick can work with glucose, but with different pathways. So different transporter, and now with the kinase, we can feed this compound directly into glycolysis, and again take away CO2 from the system. And now they're only the engineered cell is able to do this. And this is just some of the quantified data from that, looking at cells over time. So what we're hoping to do is actually give them a way to use this fixed carbon as an energy source. They can use it as a carbon source, but so far not as an energy source. And that's a little bit puzzling why that is, but I think it's just because they're autotrophs and they're not used to having a sugar to eat. But the idea is maybe if we engineer them, if we could put maybe some additional components in this pathway and get them to eat this fixed carbon, we can then use traditional genetic techniques to try to understand what the iron oxidation pathway is. Right now, their energy is completely tied to iron oxidation, but if we give them a new way to make energy, we should be able to take them off of that. And then once we understand what those pathways are, maybe we can engineer electron uptake pathway into things like E. coli or Shewanella. And there's all sorts of biotechy reasons to do that. All right, so I'll wrap that up here and thank everybody for your attention. So most of the work I talked about today was done by Bridget Conley and our collaborator, Izzy Baker, in Pete Gergis's lab. And then the Mariprofundus work I talked about was from Abhinay Jain. And on that project, we collaborate with Claire and her group at University of Delaware. And I usually throw a picture of Daniel in here lighting methane on fire. He's my friend and colleague here at Minnesota that does a lot of the work on geobacter. And yeah, good. So I would be happy to take any questions if people have them. And I will, maybe I'll stop that little screen share. I can see, there we go.
Thanks, Jeff. For some reason, my video has decided not to work, so I'm going to be a black box on my picture. But people can ask question either in person or on chat. So I see that Francois has a question. Do you want to?
Yeah, thank you very much for the very interesting and great talk. It's beautiful. I was just wondering, when you have the concatenation of the MtrA ABC or CAB, wouldn't there be possibilities that the thing could work and transfer electrons outside, having something in between the MtrA and MtrB or MtrB and MtrC? And wouldn't it be one even major case in the biodiversity, I would say?
Yeah, this is a very good point. The reason that we constrained ourselves to looking at for all three components is because we have functional evidence for those three components together in Shewanella and in Vibrio and Aeromonas. And so we were trying to narrow, I mean, there's so much stuff out there. We were trying to narrow what would come back to things that we had reasonable confidence that would really be doing this reaction. If we do a similar search with just MtrA and B, we get a ton more things out there, including, so there's a pathway that's been described as MTO in sideroxydans and some other iron oxidizers that have components that share some components that share similarity with the Shewanella system that I mentioned, in particular MtrA and B, which would be MTO A and B. There's no good genetic data, though, that those genes are involved in iron oxidation. There's been some biochemical data, and so we know a little bit about the biochemistry, but the problem with biochemistry and redox proteins is that they'll do whatever you want in a test tube as long as the redox potentials match. In the cell, though, they might not necessarily be doing that. And the case in point is some of the early biochemistry done with MtrA and Shewanella. If you artificially reduce it and throw it at iron oxide, it causes that iron oxide to get reduced. But Shewanella doesn't do that because it's in the periplasm, buried in this other protein. Like, it never sees iron oxide. So I'm always a little dubious of some of that biochemistry. And we are working with ES1 and trying to get some genetics going in that strain as well. And that guy's also been very challenging. I think one of the reasons we know so little about these iron oxidizers is that they're just a pain in the butt to work with, but they're super fascinating. We also see lots of evidence for kind of shuffling of that outer membrane multi-heme cytochrome MtrC. So in some cases, we'll see up to 5 copies of that, each of them probably doing something a little different. And we don't, and then in some cases we see an MtrA and B with something that doesn't look like MtrC at all, but still is annotating as something is probably doing something with electrons. So there's a ton of diversity out there, and I think it's again, I think we're being a little bit cautious in our interpretation to try to ground it back to the information that we have from the other systems. I think the way forward there is probably picking some of this diversity and then rebuilding it in our Shewanella strain where we've eliminated the native capacity, and then seeing if we can determine some of the functionality that way. And so that's something that we're thinking about. We haven't really jumped into it yet, but that's what we're thinking of.
Thank you very much.
Yeah, thanks for the question.
Karim, you have a question?
So Jeff, thank you very much. That was very exciting, and I guess I wrote like 15 questions, but I'll stick to 1 or 2. Just to follow up one on Francois' question. So you search for MtrA, B, and C in that order, right?
Yes.
And I was wondering if you can have you can swap the order and.
Yeah. So we did look at that a little bit too. So in the Shewanella genome, they encode 2 conduit complexes. One is MtrCAB and the other is MtrDEF. And they're actually like on the chromosome, they're encoded differently. So one is encoded MtrCAB, and the other would be MtrABC. We see some evidence for that too. I think in the tree that I showed, we allowed for both of those to be there. Let's see, in one of the pictures, I don't know if you remember necessarily, but the kind of my big tree picture, there were two big clades of Shewanella, and one of those is MtrCAB and the other one's MtrDEF. So we don't have a great handle on what DEF is really doing. We have some evidence it might be overdrive mode. Like once it begins doing iron reduction, they may make that pathway to help them do it a little bit better, but we don't have any great evidence for that. Like that's kind of our thinking on it. But I mean, it could be that one pathway is preferred under some environmental condition versus the other. Most Shewanella have got both of those copies. But then as we get out of the Shewanellas, we typically see bacteria only with kind of one of these pathways. And mostly it's this CAB, and I don't know necessarily why that gene order would matter. The one reason the gene order would matter is there is some evidence that the outer membrane, integral outer membrane protein that holds A and B together might be unstable without A in it. And so you might need to make A first and then make B. And so that might be why you have A and B transcribed in that orientation. But again, like that, there's not great evidence for that. There's some, if you dig into some of Tom Clark's papers, or if you talk to him over a beer, like he'll tell you some of those stories. Like it's not, it's stuff that people have noticed but have not necessarily kind of rigorously kind of tested. But there is some evidence that if you just try to express MTRB, like this, the cells are not happy and the protein isn't made well. But if you express both of them together, they're made quite well. And so it could be related to how that system matures. You don't necessarily like having a hole in your outer membrane is not that big of a deal because there are porins out there already. But maybe having one that doesn't have its A conduit stuck to it is a problem for some reason. I don't really know, but yeah, the order there is an interesting one, and again, it's probably telling us something about how that system works, and we just don't know what that means yet.
And so, a short related question. So, you showed that you showed like maybe events of loss or horizontal transfers of this operon, and I was wondering. So, you had that in Marinobacter, CP1 versus W62, and in some Shewanella strains. I was wondering if you had the opportunity to compare the method reduction efficiency between those strains, like the ones having it and the ones not having it.
Yeah, we haven't. I think so. It's known that the Shewanella violacea strain doesn't do metal reduction. So I think that's been tested, but I think that strain also requires high pressure to grow. So it's like one lab that can do those experiments, one or two, right? And the Marinobacter strains, what's funny is Sarah Glavin's group that works with the CP1 strain, they've been trying to put the Shewanella MTR pathway into it. And I sent her a couple of these slides a few months ago saying, hey, maybe you should just switch to one of these other guys that probably does it already. And she thought that was funny. But, I think it's, we don't have those strains on hand. It's just gazing at the genomes, which is easy to do, but it would be fun to try them. They do, that would be nice to do. Yeah, that would be good. We just haven't done it. Okay, yeah, thanks. And we see those were just a handful of examples. We've got probably 20 different examples where Bridget has gone in and really looked carefully to see, because one of the things we're looking for is maybe in one of these sequence genomes, if it's a real recent event, we might be able to see some of the transposase genes or phage proteins that might be associated with it, and that would be kind of a smoking gun for horizontal transfer, but we haven't found that part yet. But and we probably aren't going to look much more because you could do this basically forever, right? Yeah, digging into each of these genomes and trying to tell stories. But there's a time when you just got to say, okay, we got enough stuff and we got to move on.
Yeah. Thanks.
Yeah.
Any other questions? I have one while people think about it. I guess couple of very basic questions, but I want to pick one. I was kind of stuck on the slide which you showed about the speed of metal reduction between Shewanella, Aeromonas, and Vibrio. And you tried like replacing the conjugate structure from Shewanella into others, and how the recovery of metal reduction was achieved in Vibrio or Aeromonas. And you presented some hypotheses that why Vibrio has a low efficiency in metal reduction, and you linked it to something about survival or nutrients. The thing I was thinking is like, is this something also related to the environments in which they are originally found at a more abundance, and those environments are like not rich in certain elements which they need to acquire, or is there?
Yeah, that's a good question. I mean, so oneidensis comes from the sediment of Lake Oneida, which has I don't know that it has an unusually high amount of things like manganese and iron in it. Although I think those sediments had a fair amount of kind of toxic metals because of there was a bunch of industries that were like basically dumping into that lake, before it was not legal to do so. The Aeromonas hydrophilus strain is one that people study like as an opportunistic pathogen, and I think that was actually isolated from a person. The Natrogen strain was isolated from like a salt marsh, kind of a brackish water marsh. So I don't know that we know enough about exactly where they came from, or if they are in very iron-rich ecosystems. I would say, I guess, for these specific cases, they're not coming from ecosystems where there's a ton of iron necessarily. So yeah, I mean, it could be related to that, but I think at least in the case of Shewanella and Aeromonas, if we in lab conditions where we can give them as much iron as we want, we can see. I mean, we see these very different rates of iron reduction compared to Vibrio. So Aeromonas is about about half as fast as Shewanella, and then Vibrio is maybe half again as slow, or maybe even more than that as slow. Yeah, but I mean, ultimately, I think these pathways are operating with the efficiencies that that benefit the cell, right? In whatever environmental condition that would be. For Vibrio and Aeromonas and Shewanella for that matter, I think this metabolism primarily benefits them if they find themselves in the sediment and there happens to be some oxide minerals around, and in that sediment, goes anoxic. And if that happens, these guys can move electrons and do better than their neighbors around them that can't. So I mean, I think ultimately that's the selection for it. Yeah, I mean, and it could be that Shewanella finds itself in that sort of ecosystem more frequently than the others. Yeah, but it's a great question that we just don't really know the answer to. But I think, but we could know at least some of those answers through some additional experimentation. So one of the things we really want to do is rebuild all of these pathways in their entirety in Shewanella. The data that I showed is just the outer membrane conduits that had been rebuilt in Shewanella, but we can rebuild the entire pathways. It's just a matter of it starts getting a little bit more complex where we've got to put things into the chromosome rather than on plasmids, but we know how to do it. It just takes time and money.
Okay, thank you. Did I see someone? Francois has another question.
Yeah, I was really fascinated by what you showed at the very end of the talk about enhancing the growth rate by giving some assimilated carbon to an autotrophic carbon autotrophic strain. Your model behind that, your thinking model behind that, is that the ATP that would be produced during the iron 2 oxidation would be reallocated to growth, right?
What we were originally trying that didn't work at all was we were trying to get them to grow heterotrophically to get their ATP from breaking down these fixed carbon sources, and it never worked. And as soon as we asked a more simple question, could we just replace the carbon? then things started working in that system. And
Perfect.
and so I think that it's a specialist. Like, this organism really does one thing well, and when we ask it to do some other things, it's just, it's not used to doing that. And the electrons flow in different ways. And I think it's going to require some more creativity to try to get these guys to use this carbon. There's some really cool work that has been done recently, I guess over the last 5 or 10 years, in E. coli, where they've been trying to build E. coli into an autotroph, and they run into very similar sorts of issues, right? But from the, E. coli wants to send this carbon all over the place, and it wants to do this, but you really have to constrain it and focus it in order for it to begin working. And that's been just some heroic work that's been done there.
Then the question one could have would be, why is this xylose, right? Because it would be better maybe for the cell to use something easier, right? And it work better with something easier, right?
Yeah. So initially, when we thought of this line of experiment, I guess we expected that it wasn't just going to work, right? That we would give them this pathway, and so what we had proposed was that we would evolve those cells, basically continuously grow them with this new capacity, with the carbon source present. And if one rare cell figures out how to make a living that way, it would take over the population quite rapidly, right? And this is called, this sort of evolution experiment is done all the time in E. coli and Shewanella and Geobacter, for that matter. The problem is with Mariprofundus is that it grows slow, and we never get very many cells. And so those evolution experiments become exponentially more difficult because we don't have the numbers. And so I think we have to be smarter about how to kind of think of what those bottlenecks are going to be, and try to give the organism solutions to those bottlenecks, because I think we just can't depend on getting enough numbers for the evolution aspect to really help us. But we thought that would work. And yeah, we had a reviewer of one of our proposals who ended up, like, this funded up one of my postdocs, Abene, to work on this for a while. But this is Steve Finkel at USC, who thinks an awful lot about evolution. He knew the numbers were not probably going to work out. But yeah, he, as usual, he turned out to be right, I think. But maybe, if we can head off maybe 3 or 4 other bottlenecks, and not demand that many solutions be solved by evolution. Maybe there's 1 or 2 things that it could solve for us, with some transfers. I mean, ideally, maybe what we really need to do is just get a chemostat running, and just let them go, and just, keep an eye on how things are working. And at some point, if we see the carbon just plummet or the cell biomass start increasing, then we know something's happened. And maybe 4 or 5 grad students down the line, in a few years it would happen. I just don't know. But yeah, maybe like a long-term chemostat experiment would be the way to go. Yeah.
Thank you very much.
Sure. Great question.
You have a question?
Yeah, I may have a last one if you allow me to. So you showed that Shewanella versus Aeromonas versus Vibrio were reducing metals at different rates, and I was wondering, so when you were comparing the whole protein set of those strains, can you replace so that was I noted that down, the CCTA and FCCA of Shewanella by the PDSA of Aeromonas or Vibrio to see if that's.
So we have done those experiments, and they work just as fast in the Shewanella background. But that would be with we've done that experiment, but only with the Shewanella conduit. So we have done all the swappings except the entire pathways. So we haven't moved the conduit, the periplasmic, and intermembrane components all at once, and that's really what we want to do. But that experiment makes me think it's something about either Shewanella's ability to make multi-heme cytochromes, or just something about its metabolism, that it's routing electrons this direction under these conditions more robustly. We do know that that Shewanella does make lots of multi-heme cytochromes there. If you grow them anaerobically and give them enough iron, like to make them happy, that the cell cultures are very pink, and the colonies are pink on a plate, because they're making so many multi-heme cytochromes. That's not true with Aeromonas and Vibrio. Like they don't make, just visually on colonies on a plate, like they're not pink like Shewanella. So like we know that they don't make as many multi-heme cytochromes as Shewanella can do. And so that could be the difference. In order to answer that, then if in our Shewanella background, where we've removed everything, we do have some expression systems where we can control the levels of expression. And so the prediction would be that we would, if we can figure out kind of the levels that Vibrio normally makes, if we tune that system in Shewanella to make that amount, maybe we'll get Vibrio rates directly, right? Or if we tune it up to Aeromonas levels, maybe we'll get more versus Shewanella levels, we'll get the most. And I think that might make sense. It still goes back to the questions that Neha asked about kind of, what's the environmental driver, the selective pressure to have these systems operating at the different levels. And I think that's a really interesting thing to understand, and I think a really challenging thing to understand. But that might be the best way to even begin, making predictions about what might be happening in these other systems too.
Thanks, that's very clear.
All right. I don't see anything in the chat. Neither do I see any hands raised. So since I have 1 more minute, I'm going to ask a quick question, Jeff, before we end the seminar. So you talked all about iron. And in the beginning, you showed a lot of terminal electron acceptors which can be utilized. So the most common cousin of iron is manganese. The kind of pathways you showed and the efficiency in reduction, is there a reason to think that these efficiencies and the pathways would differ depending on the electron acceptors available?
Yeah, that's a good question too. So a lot of so I showed that big list of electron acceptors that Shewanella can use. Almost everything on the right-hand side, all the metals and things are going through this pathway. Okay. So some of them, I get the arsenic is not, that's a little bit different, but most of that metal reduction we think is linked to the operation of this pathway. And at least from what we can tell, iron and manganese oxides are both reduced through this pathway. Now the question about efficiencies or speed, I mean, the speed stuff is really coming from experiments with chelated iron 3, with iron citrate only. We've not done those experiments with iron oxides or manganese for that matter, and those would be good things to do. But I wonder if with the insoluble substrates we begin, there ends up being a whole bunch of physics and things and diffusion that ends up confounding rates in a way that we don't see with iron citrate. But from that same perspective, I wonder, I mean, the more these bacteria are unlikely to run into 5 millimolar iron citrate in the environment, right? They're more likely to find 5 millimolar iron oxide or manganese oxide, something like that. So yeah, I mean, I think in general what we've noticed, looking at the overall rates of reduction, that what we see with iron citrate is mimicked with iron oxide and manganese oxide. But like those experiments in the Vibrio system, I'm not even sure. I think like that experiment went weeks and weeks and weeks. And once if an experiment's taking a month, I'm less interested in it. Yeah, but that is something that we should keep in mind, that we are giving them like basically an ideal situation or scenario with a chelated iron 3 molecule that we know is reduced on the outside of the cell, based on older work that we've done in Shewanella. But yeah, it doesn't necessarily reflect what these guys would normally be seeing out in reality. That's for sure.
Yeah, cool. All right, I think we're run out of time, so I'm gonna end the seminar. Thank you so much, Jeff. It was a wonderful talk, really informative. And I can say that I don't know anything about genetics or anything or any kind of sequencing. I understood it, so
great.
So I think it was great.
Great. Well, thanks for that.
Yeah, thank you for the talk, and then have a good rest of your day. And thank you everyone for joining us, and see you next month for next set of talks.
Yeah, thanks very much. It was really fun. And Kareem, Niha said you're going to send me a bunch of French wine. Is that right?
Did I say that? Okay, I can work on it.
Next, when we get to do conferences again in Rio,
Yeah, we'll drink it
have a
together.
beer and some wine.
Well, who knows? Maybe you're coming to Ghostbed. Oh.
Okay, thanks everybody. Take care.